Hello, I'm Antoinette Adams, and today I will be presenting an informal PowerPoint on glycolysis, gluconeogenesis, and the role of autolyte gene expression. I am currently in biochemistry, and this is my final presentation. Today, we will learn glycolysis and the gluconeogenesis pathway overview. We will discuss how autolysis is involved in both glycolysis and gluconeogenesis. We will learn different isoforms of autolysis and their expression patterns. And we will also explain how autolyte B expressions can be regulated by transcription factors. And we will discuss 3 to 4 of them. So, a quick overview on what we'll be discussing today. Glycolysis is a process that allows cells to anaerobically extract a limited quantity of energy from glucose. So there are two phases. You have the preparatory phase, which consists of 5 steps, and then you have the payoff phase, which also includes 5 steps. A process called gluconeogenesis allows cells to utilize a range of metabolized and synthesized glucose. The distinctions between glucose production and glycolysis, and we will discuss how they are both rendered, and we will also discuss how they are independently controlled to prevent a various cycle. We will also describe the pathways that brings carbs onto the glycolysis and other hexes, disaccharides and polysaccharides. All living things typically have a metabolic process called glycolysis that occurs in the cytoplasm of their cells through the conversion of glucose and pyruvate, ATP, and NADH. Two forms of energy are released and reduced respectively during this process. So there are a total of 10 steps that make up glycolysis, and each one is catalyzed by a different enzyme. The steps can be divided into 2 phases. Once again, you have the preparatory phase, which are steps 1 through 5, and then you have the payoff phase, which are steps 6 through 10. Glucose is phosphorylated to glucose 6 phosphate by hexokinase. Glucose 6 phosphate is isomerized to fructose by phosphoglucose isomerase. Step 3 is fructose, which is when the phosphate is phosphorylated to fructose. You have step 4, which is when the biphosphate is split into 3 carbon sugars by autolysis. Step 5, the 3 carbon sugars are summarized by glyceride, phosphate by triose, phosphate isomerase. So I know you're probably wondering, why is glucose a fuel that is needed so frequently? It usually exists in a stable ring shape and doesn't usually glycolate proteins produced under circumstances from formaldehyde. So the second phase, those are all the steps of the preparatory phase. So then you have the second phase, which is payoff. The first step of the payoff phase would be glyceraldehyde. That is when phosphate is oxidized and phosphorylated to steps 1 and 3, where the biphosphorate dehydrogenase. Then you have the second step of the payoff phase, which is when biphosphorate donates a high energy to phosphate to ADP, which eventually forms ATP. Third step, you have phosphoglycerate kinase. phosphoglycerate is arranged to 2-phosphoglycerate by mutase. Then you have the fourth step. Phosphoglycate is dehydrated to phos- phosphorpyruvate, and that's activated by enos. Then you have the last final step, which is phosphorpyruvate donates a high energy to phosphate to ADP, which forms ATP, and pyruvate by pyruvate kinase. And so gluconeogenesis, on the other hand, is a metabolic pathway that generates glucose from non-carbohydrate carbon substrates such as lactate, glycerol, and gluconeogenic amino acids. It is essentially the reverse of glycolysis with a few key differences to bypass the inversible steps in glycolysis. The enzymes that catalyze these bypass reactions are pyruvate, carboxylase, and phosphoryl. Here is just a visual of the preparatory phase that includes those first 5 steps. So here we have glycolysis, an actual breakdown of it. Glycolysis is a mechanism that produces a tiny amount of energy from glucose and converts it to either lactate or pyruvate breakdown. Can't see the rest of that. So all the intermediates in glycolysis have either 3 or 6 carbon atoms. All of the reactions fall into one of 5 categories. Either it's a phosphoryl transfer, it's a shift, it's been isomerized, it has become dehydrated, or an idol cleavage. It is the sole route that functions in every cell in the body. The only energy source found will be in the brain, sperm, and the renal medulla is glycolysis. During intense physical activity, anaerobic glycolysis takes over the primary energy source for muscles due to a shortage of oxygen in the tissues. One could think of the glycolytic pathway as the first stage leading up to total oxidation. The carbon skeletons for the synthesis of non-essential amino acids and the fats glycogen component are provided via the glycolytic process. The majority of the reactions can be reversed. Of what value is glycolysis for cells? One, the value for glycolysis for cells will be that it provides energy in form of ATP. It provides building blocks for synthetic reactions. And I know you're not asking, like, so what are the most control points found? Well, enzymes that catalyze irreversible reactions would be hectokinase, phosphorufructinase, and pyruvate kinases. Or kinase. Here you have pyruvate, decarboxylase. Yeast contain the enzyme pyruvate that decarboxylates pyruvate to form uh, acetaldehyde. That is then reduced to ethanol under certain conditions. So you have the anaerobic glycolysis, which is NADH produced, cannot be used by ETC for ATP production. So the less ATP production as compared to aerobic glycolysis. Lactate is an obligatory end product. Why? Because if it is not formed, all cellular NADP plus will be converted to NADH with no means to replenish the cellular NAD. So the glycolysis stops, and then you have the death of the cell. An essential enzyme in the metabolic process is aldolase. Its function in glycolysis is to divide fructose by phosphate, a 6-carbon sugar, which turns into 2,3 carbon sugars. So this is an important phase because it prepares the body to harvest energy from the glycolysis in the form of ATP in later stages. Autolysis catalyzes the opposite reaction during the process of gluconeogenesis, which is the synthesis of glucose. So you have fructose, 1,6-biphosphate is created by combining dihydroacetone phosphate with glyceraldehyde phosphate. This is a crucial point of regulation for the gluconeogenesis pathway, since it's one of the few gluconeogenesis steps that is not just the opposite of the corresponding glycolysis phase. In both pathways, autolysis plays a key role in the regulation of glucose metabolism, and this activity can be influenced by various factors, including the availability of substrates and the energy needs of the cell. Here you have the phase of pyruvate generated by glycolysis. Pyruvate is just the end product of glycolysis, which can undergo various fates depending on the cellular conditions. Not just depending on the cellular conditions, but also its energy demands and the metabolic pathways active within the cell. So you have conversion to acetal. Pyruvate can be converted into acetal CoA through a reaction catalyzed by the enzyme pyruvate dehydrogenase complex, which is PDC. This conversion occurs in the mitochondria and is a crucial step in linking glycolysis to the citric acid cycle, also known as the Krebs cycle or the TCA cycle. So acetal CoA enters the TCA cycle, where it is further oxidized to generate ATP through activized phosphorylation. The second, you have lactate fermentation. Under anaerobic conditions or during intense exercise, when oxygen availability is limited, pyruvate can be converted into lactate through the enzyme lactate dehydrogenase. This reaction regenerates NAD plus from NADH, allowing glycolysis to continue in the absence of oxygen. Lactate is then transported out of the muscle cells and can be used by other tissues or converted back into pyruvate by the liver through the Cori cycle. You then have the ethanol fermentation, which is yeast, and it contains microorganisms. Pyruvate can undergo alcoholic fermentation, where it is converted into ethanol and carbon dioxide. This process involves decarbonization of pyruvate to acetaldehyde by enzyme pyruvate decarbylase, followed by a reduction of the acetaldehyde to ethanol by alcohol. So ethanol fermentation is important in the production of alcoholic beverages and in industrial processes like bread making. And when you create bread, you're also utilizing there's yeast as well. So then you have the gluconeogenesis, which is when the pyruvate can also serve as a precursor for gluconeogenesis, the synthesis of glucose from non-carbohydrate precursors. In this pathway, pyruvate carboxylase converts pyruvate into isoacetate, which is then converted into phospholipid pyruvate, PEP, by PEPCK. So glucose is subsequently synthesized from PEP through a series of reactions. Gluconeogenesis is vital for maintaining blood glucose levels during fasting or periods of low carbohydrate intake. You then have the amino acid synthesis. Pyruvate is an important precursor for the synthesis of certain amino acids. For example, you have alanine, which is synthesized from pyruvate through a transamination reaction catalyzed by alanine amino transferase. Moreover, however, pyruvate serves as a precursor for the synthesis of other amino acids, such as serine and glycine, through various biosynthesis pathways. You also have fatty acid synthesis in lipogenic tissues, such as the liver and adipose tissue. Pyruvate can be converted into CoA, which serves as a precursor for fatty acid synthesis. CoA generated from pyruvate can enter the cytoplasm, where it undergoes carboxylation to form malonyl CoA, which is just the building block for fatty acid synthesis. So when you mix and combine, the pyruvate, the face of pyruvate that are generated by glycolysis, you will see that they all illustrate, a central role in cellular metabolism, serving as a key junction point between glycolysis, the citric acid cycle, gluconeogenesis, and various biosynthetic pathways. The fate of pyruvate is tightly regulated by cellular energy demands, substrate availability, and metabolic interconnections to ensure the proper functioning and homeostasis of the cell. Additional information: an enzyme called aldolase is essential to the metabolic process known as glycolysis, which turns glucose into pyruvate. You have three isoforms that exist in humans. You have aldolase A, aldolase B, and aldolase C. Each isoform is encoded by a different gene and expresses itself in various organisms with unique expression patterns. Aldolase A is just tissue distribution. Aldolase A it's primarily found in muscle tissues, including skeletal and cardiac muscle. You have the function. Its main role in glycolysis, where it catalyzes the reversible conversion of fructose biphosphate to glyceraldehyde phosphate and dihydroacetone phosphate. You have the expression pattern. Expression of aldolase A is highest in tissues with high metabolic demands, such as your skeleton muscle. Your skeletal muscles is where it is very important and very essential for energy production. You then have aldolase B, which is the tissue distribution. It's predominantly expressed in the liver and to a lesser extent in the kidney. You have the function. Its primary function in the liver, where it catalyzes the cleavage of fructose phosphate into glyceraldehyde and dihydroacetone phosphate. This reaction is essential for metabolism of dietary fructose. Its expression would be mainly in the liver. Its expression pattern would be expressed in the liver. The liver plays a crucial role in fructose metabolism. Its expression is also detected to a lesser extent in the kidney. You then have aldolase C. Its tissue distribution is found in the brain and the nervous system. Its function catalyzes the same reaction as aldolase A and is involved in glycolysis in the brain, where it contributes to energy production. Its expression pattern is highly expressed in the brain, particularly in neurons and your glial cells. Its expression is crucial for glucose metabolism in the central nervous system. The different expression patterns of aldehyde isoforms reflect, their special roles in distinct tissues and metabolic pathways. So, while aldehyde A and C are involved in glycolysis to provide energy for muscle and brain function, aldehyde B is primarily responsible for fructose metabolism in the liver. These isoforms demonstrate tissue-specific regulation to fulfill the metabolic requirements of different organs and maintain overall metabolic homeostasis. Now you have signals, the energy states, the availability of biosynthetic intermediates, and then you have the metabolic state of the whole organism. So regulation of metabolic pathways, the flow of intermediates through metabolic pathways is usually controlled by four mechanisms. The first mechanism would be the substrate availability. The second would be the allosteric activation or the inhibition. You have the covalent modification of enzymes, and then the induction / the repression of enzyme synthesis. It's important to know that metabolic regulation of biochemical process pathways makes psychological sense. Control of flow-through pathways often distribute among several enzymatic steps in the pathway. Here is a visual that shows us G6P. Fate depends on tissue. In muscles, the GP6 proceeds through glycolysis. In the liver, GP6 is converted to glucose. So this shows us from the G6P eventually converting to glucose at the end, which is blood for use by other tissues. It allows your other tissues to use the blood. Lastly, we have the transcription factors. So different transcription factors can bind to distinct regulatory regions in the promoter region of the aldehyde B gene and control the gene's expression. Transcription factors are proteins that bind to specific DNA sequences and control the transcription of genetic information from DNA to mRNA. They function by promoting or blocking the recruitment of RNA polymerase to specific genes. So there are a few different transcription factors. The first one will be HNF1, which is hepatoside nuclear factor. So this transcription factor plays a very important role in simply regulating the expression of numerous liver-specific genes. It binds to specific DNA sequences known as HNF1 binding sites within the promoter region of the gene. The activation of HNF1 leads to the transcriptional activation of aldehyde B, resulting in increased expression of aldehyde B in liver cells. So HNF1 regulates various aspects of the liver function and metabolism, including glucose metabolism and the expression of genes involved in glycolysis and gluconeogenesis. You have the second transcription factor, which is HNF3. It's known as the forkhead box A, which is also known as FOXA. It's another transcription factor involved in the liver development and function. It binds to elements within the promoter region of aldehyde B gene. HNF3 can act synergistically with HNF1 to enhance the transcriptional activation of aldehyde B, furthering promoting the expression of aldehyde B in hepatocytes. And so just like HNF1 and HNF3 plays a crucial role in the regulation of liver-specific gene expression in the metabolic pathways. You then have CCAAT, or the enhancer binding protein. The enhancer binding protein is a family of transcription factors that regulate various cellular processes, including metabolism, the differentiation, and immune responses. Certain isoforms are expressed in the liver and can modulate IDLA B. So CEBP binds to CEBP binding sites within the promoter region of IDLA B gene and can either activate or repress its transcription. It just depends on the cellular content and the presence of the cell. So CEBP is involved in the regulation of gene expression during different psychological states such as fasting, feeding, and metabolic stress. You then have the D-box binding protein, which is just a transcription factor. It belongs to the part B-zip, which is the proline and acidic amino acid rich basic family. It regulates gene expressions in a circadian manner, playing a role in the circadian rhythm of gene expression. So DBP binds to D-box elements within the promoter region of IDLA B gene and can influence its transcriptional activity. The circadian regulation of IDLA B expression by DBP contributes to the rhythmic variation in IDLA B levels in the liver, which is coordinated with other metabolic processes to optimize energy metabolism throughout the day. These transcription factors integrate various signals and metabolic cues to control the expression of IDLA B in a tissue-specific and content-dependent manner, thereby contributing to the regulation of glucose metabolism and other metabolic processes in the liver. There you have it. I hope that you were able to gain insight and some additional information on gluconeogenesis and glycolysis and IDLAs. My name is Antoinette Adams, and it has been a pleasure presenting to you. Thank you.